Hi, my name is Leah Chen, and I will be presenting my research on a novel hydrogen peroxide assay technique for characterization of hydrogen peroxide responsive nanoparticles. AP103 is a newly developed nanoparticle treatment for cardiac surgery-associated acute kidney injury, or CSA-AKI. The nanoparticles are composed of a PVAX core, which are the active ingredient, and a PVA shell. The PVAX reacts with the reactive oxygen species, ROS, the cause of CSA-AKI, and releases vanilla alcohol, an anti-inflammatory. In preparation for clinical use, a hydrogen peroxide assay is necessary to determine the remaining hydrogen peroxide after contact with AP103. Isothermal titration calorimetry is a promising new assay that can offer more quantitative information than the assays that are currently in use. The goal of this project is to fine-tune the process of quantifying the heat reaction between AP103 and hydrogen peroxide using ITC. Acute kidney injury is a common cardiac surgical complication. AP103 consumes the reactive oxygen species to prevent future injury, and the anti-inflammatory that is released treats the site of injury. For the patient, this means shorter hospital stays and a quicker recovery. Preparation of the particles is done with a high-shear mixing of PVAX and dichloromethane and PVA and water. The two immiscible solutions form an emulsion where the PVAX is encased in a PVA shell. Rotary evaporation is used to remove excess dichloromethane, centrifuge rinsing techniques are used to remove excess PVA, and water is removed through freeze drying. The current method for quantifying the reaction between AP103 and hydrogen peroxide involves changing the concentrations of AP103 dispersed with hydrogen peroxide. After 24 hours, the leftover concentration of hydrogen peroxide is measured using a Pierce quantitative assay kit. This method only quantifies the amount of hydrogen peroxide that is consumed by the reaction and fails to detail any reaction kinetics. Isothermal titration calorimetry is a technique that reveals the thermodynamics of a reaction. In a slow and controlled titration, the heat released is measured during each increment of time. ITC quantifies the kinetics of the reaction more than the current assay by providing data on the reaction rate and gradual consumption of hydrogen peroxide. The aim of this project is to identify the ideal concentrations and parameters for the titration such that the AP103 reacts fully with the hydrogen peroxide and reaches zero heat of reaction at the conclusion of the titration. Next, I will show the methods used in this titration. First, a sample is made and degassed to prevent any air bubbles that might pop and release heat during the titration. Next, the hydrogen peroxide is placed into the ITC. The titration needle is carefully filled with the AP103 solution. The top is tapped to remove air bubbles, and any solution that escaped is refilled to the proper amount. The needle is tapped gently to make sure that no air is trapped in the tip of the needle either. The needle is screwed into the handle carefully, and the handle is placed into the ITC. Settings regarding time increment are specified, and now equilibration can begin. Here, we can see a comparison between my results and the ideal results that would be displayed if I had met my goal. Compared with the ideal titration results on the bottom, there seems to be a buildup of background heat in my trials, which indicates that some parameters must be adjusted. There are several factors that can impact the quality of results. The heat of air bubbles popping can be resolved through degassing. pH differences between the AP103 solution and peroxide solution can be resolved through using low concentrations of each compound, as well as verifying pH levels before running the test. Temperature is another factor, but this is usually mitigated by preparing the samples in the same lab setting. Lastly, background heat simply from the peroxide into water reaction is inevitable. Although the experiments have not yielded conclusive results, progress has been made in working with ITC in the context of quantifying this chemical reaction. We have successfully identified factors that can affect the results, which will be tested in future experiments. These results will help fine-tune the parameters and hopefully establish a more efficient, quantifiable method for characterizing chemical reactions.